Good morning. Today, I would like to share with you these dynamic flow and shear stress as key parameters for intestinal cells morphology and polarization in an organ on a chip model. As you know, microfluidics has enabled to recapitulate the physiological conditions that cells face when they are exposed to perfusion. So in this research, the authors developed a gut-on-a-chip model. As you can see here, this is the architecture of this microchannel where the cells are going to be seated. The cell line that they use is colon carcinoma, CaCO2 cell line. And here you see how they culture the cells in this gut-on-a-chip device, which is connected through tubing to this peristaltic pump that delivers the fresh media through the inlet and also makes possible for the spent media to go out through this tubing to this reservoir. And everything is placed inside this incubator at 37 degrees Celsius, and it has a CO2 supply 5%. Here you see an schematics of the experimental design. So the first day, they seeded the cells, so they are rounded, and after 1 day, they are attached to the channel. So they start pumping. And polarization starts. It means that the morphology of the cells changes from rounded to they become polarized, so they become higher. And they, at day 5, they change the flow rate, and at day 8, they stop the pump. So how do they correlate this stress caused by the flow to the morphology of the cells? So they use computational fluid dynamics simulations, and they apply this formula in order to calculate the flow rate that they needed to apply to for a shear stress value of 0.029 per centimeter square, which is reported for cell polarization in this specific cell line. And so they use that flow rate value and for the simulations, and they calculate the cell shear stress. So here you see for the small cells, the medium and tall cells, these values of the cell shear stress. You can see how the morphology of the cells changes and according to its height and width. And they related the shear stress. The shear stress was the main cause for the cells to polarize and to form this microvilli architecture, which is main feature of these intestinal cells. And they also used matrihel in order to represent this extracellular matrix, and they discovered that in the static culture, the microvilli were not observed. That's why they conclude that this microvilli architecture is enhanced by the flow rate. So it doesn't depend on the matrigel extracellular matrix. So here you see how the microvilli architecture was obtained when the cells are exposed to perfusion, and it doesn't appear here when the cells are cultured in a static mode. Okay, so here you see the C represents constant flow rate, and D dynamic flow rate, and S static. It means no flow rate. Oh, right. So they also measured the genetic markers in order to see if they were altered by the dynamic flow rate. And here you see a comparison with this housekeeping gene and the expression of other genes, how it varies according to this constant flow rate, dynamic flow rate, or static flow rate. And you can see that the gene expression has not been altered. Also, they wanted to see how the shear stress influences 3D organization and mucin expression. So they noticed that F-actin expression um, it changes according to the flow rate exposure. Here you see these confocal images for the constant and decreased flow rate conditions on day 8. And this is the merge photograph for this protein Z01 or bilin. The nuclei is in blue, and how it is proof that the properties as the barrier are maintained and are enhanced by this perfusion. So also there was this theory that stated that cells were compared to Backlage. So they also noticed that this theory is able to be applied for small cells. But in the case of cells that are tall, this theory doesn't work anymore. So an important um, um, conclusion of this study is that for the tall cells, the bulge-like theory doesn't apply. And this is why they combined these simulations and in vitro microfluidic culture, and they were able to model and establish this optimal shear stress for this cell line in this gut-on-a-chip model. So previously it was reported that the optimal shear stress was 0.02 dyne per centimeter. And from the simulations, they could estimate the optimized one for tall, and it was about 0.05 dyne per centimeter. And that's how they predicted this cell shear stress. All right, thank you very much.